Hi, we're here with Mary Hagedorn of the Smithsonian Institution, and Morris Animal Foundation has funded one of Mary's grants to study reproduction in coral. Mary, could you tell us a little bit about coral and why they're so important?
I think it's really important that people understand that they're animals and not plants or rocks, as many people think they are, and that they're really important to our ecosystems, and their health is really important to our ecosystem's health. Morris Animal Foundation is funding a program that I'm working on to try and conserve endangered coral, in particular the Acropora palmata, which is called the elkhorn coral and lives mostly in the Caribbean. This species at one time dominated the Caribbean, and it's a massive, they call it reef building coral, and it covered most of the ocean in the Caribbean, and is now being severely threatened by diseases and other sort of forces or impacts that are causing the populations to contract. What Morris Animal Foundation has been interested in is ways to conserve this coral species that include ex situ conservation, or it's called out of the place kind of conservation. And we're using human fertility techniques to freeze the sperm of Acropora palmata, or the elkhorn, and we're then banking that sperm to then potentially use this sperm to diversify populations that are constricting, like some of the populations in Florida. We're also identifying cells that might be useful in developing cultures for coral, like stem-like cells in coral that could potentially be used to analyze disease in coral.
And so what are some of the things that you've done so far, and how do you see those impacting the coral going forward?
With Acropora palmata, we have been able to successfully freeze down the sperm from this species. And we've also banked this sperm in a number of international germplasm repositories. We are hopeful that we can gear up this now sort of small project to then use in a larger conservation way, so that we can freeze enough sperm that it could be used to fertilize fresh eggs and be used to diversify populations. We're not currently doing any of that work now, but we're preparing for the future when these sorts of demands might be needed. And for the stem cell work, we are really in the very beginning stages of that work, trying to analyze their properties and dissociate those cells, potentially cryopreserve them, and we are collaborating with people who might be able to culture those cells and grow them up.
Okay, and just the last question is, what do you think is the most important thing that maybe people need to know about coral and about this study, and kind of what they can do?
Those are really good questions. I think it's often frustrating for people who do want to do something. I think the best thing that people can do is they can contribute to places like Morris Animal Foundation, because often people can't get to the Caribbean, or they can't volunteer for a project like mine, but supporting foundations like Morris Animal Foundation, which helps scientists, help wildlife, is really crucial. I think that what my project is doing is it's opening a small door in another technique to help save coral. You can do in situ conservation, which where you might help form a marine protected area, but there's lots of other things that we can do, and my project is actually forming an insurance policies for coral for the future, and it's now just being done on a couple of species, but we're hoping that what we learned from the elkhorn coral and other species that we can apply more broadly and help train other people throughout the world to do the same sorts of techniques. And so there'll be lots of gene banks with lots of coral from all over the world.
Great. Thank you so much.